Hello, hello. Can one of you guys do me a favor? Because I realized I'm getting close to running out of time. Can one of you, like, get me some water? Because I'm about to be, like, shouting forever. What is what again? Kids pick cooties off fat Girl Scouts. Yes, ma'am, indeed. But yeah, this one I think is going to be a lot shorter. Review, yeah, because he was here for the other review that I did. Like an hour ago? Like for chemistry. For what?
There's only 41 questions. There's still 41 questions.
That's not the one that I'm doing, actually.
But it
I
has
know.
your name on it.
I know.
It's the one that was
I know.
on the board.
Thank you.
On the website.
Well, because there's been a change and...
So you changed the one on the website?
I did.
I printed these like 3 days ago.
I know. I submitted a revision to them, and it's the same thing, but I eliminated a couple of questions.
Oh, so this is fine.
It's fine, minus a couple questions. Thank you. Is the microphone good? All right, cool. Okay. Okay, can I have your attention, please? Hello, one and all, and welcome to this, our final exam review for Biology 1. Can everyone hear me okay? Can you hear me now? No? I'm not using, okay. So here's the situation. I'm going to illuminate this for you. This microphone is very loud. So here's what happens when I use it. All right, it's going to kick on in a second. Yeah. Okay, so we can try it like this, but I may end up getting rid of it because it's incredibly loud and it makes me sad, and I tend to get enthusiastic. So we'll see how this ends up going. We'll go ahead and try it with the microphone. Can you hear me okay? All right, so I'm assuming that you can. Okay, so we're going to go ahead and get started. I have a couple things to say. First off, there has been a revision to this exam review. So if you are operating with the one off of the Special Study Union website, it's the same thing. I just eliminated a couple of questions. You can find the most updated version on my personal Google site. So if you go to Start Google site, Bio 1 with Jessica, that's where the most current one is. You guys are also welcome to get pictures of anything that I have. Whew. Yeah, it is on. Sorry about that. Okay, I guess they're trying to figure this situation out. In the meantime, I'm just going to ask that you guys do the best that you can to listen to the words that I have to say. and we'll have a good time. We have, I believe, 32 questions. Yeah, 32. So that's the final cut. Because they took some stuff out of Bio 1 and moved it to Bio 2, which is great news for you because that means that you don't have to learn it. So yay, we're excited. Although Hardy Weinberg is kind of fun, so I'm a little bummed, but that's okay. I know you're like really disappointed. But anyway, a couple other things. So if you notice up here, there's this code thing, and it's on the tables actually that you're sitting at. There's a couple of them posted around. So back in the olden days, they would hand out, they meaning like I at your session, we'd hand out pieces of paper asking how you felt about the session, how you feel about study union. They ended up making a change because they didn't want to waste a bunch of paper. However, it's still really, really important that we do get feedback. It helps make this, it helps keep this program alive. So please, at the end, if you have a moment, do scan one of them with your phone and fill out the survey. It's real quick if you're willing to do that for me. But yeah, this is our last moment with Bio 1. It's been a really awesome time, and I think we're going to have a fun time going through this. So if you have any questions at any point, please let me know. We can definitely do more. I just tried to hit some highlights, okay? So we'll go ahead and get started. Do you guys have any questions before we begin? You? You want my sweater? I'll totally give it to you. Yeah. No, I'm serious. Okay, so we're going to go ahead and get started. Okay. Yeah, I did have it on earlier, and then like you start moving around, and you're like, okay, I don't need it anymore. You could run this with me, you won't be cold. Okay.
I replaced the batteries in this one, so
It should be
it should be good. I turned it down a little bit too. It was pretty
okay. Yeah, it was really loud.
Yeah, I'll stay in here for a couple minutes.
Okay. Is it on?
No.
No?
Yeah, go ahead and turn it on.
Hello, hello? Hello? Woo, okay. All right, I'm still gonna keep this over here. Okay, I think that sounds all right. Yeah, thank you. I appreciate it. Okay, so we do have a microphone. It's gonna be a little bit quieter for us. All right, let's go ahead and get started. We're gonna go back to the time before time, to chapter 1, okay? So we're looking at one of these comparison problems where we have species that are in the same blank or more closely related than those that are only in the same blank, okay? So what do we need to be able to compare to do this? What are we going to be looking at? You guys remember that whole taxonomy thing, right? So we got kingdom, phylum, class, order, family, genus, and species. There's a couple fun ways you can remember it. One of the more traditional ones is King Philip came over for great spaghetti. There's one that I heard that's bizarre yet memorable. It's kids pick cooties off of fat Girl Scouts. I don't know why, but it fits the letters. If you have an alternative and it works for you, then by all means, please go for it. But the idea here, the thing that's the most important to remember is not just the order, but what the implication of the order is, okay? So what this means is that as we go further down, we get more specific. You guys remember doing this? So we get more specific the further down we go, because if you think about it, like we as humans, we are Homo sapiens, right? That's our genus and species. But we are from the kingdom Animalia. There's lots of other animals. There's giraffes, there's slugs, there's fish, there's birds, right? All these things that are animals, but we are only one species. All right, so you see how this kind of goes? The more specific it gets, the more closely related you end up getting, right? Because we're going to be more closely related to other primates than we might be to, like, worms, right? So, or like bananas, which is a plant. It's actually really interesting how closely related you are to bananas. You should, like, look that up. But in any case, okay, so in any case, let's try and answer this question given that knowledge, okay? So what we're saying is species that are in the same something are more closely related than those that are only in the same something else. So we're looking for, if it's going to be more closely related, it's going to be lower down on the list, okay? So those in the same phylum are not closer than those in the same class. The lower down you go, the more specific it gets, the more like intimate it becomes. Okay, so this one is not going to be our answer. Then we have family and then order. So this is an interesting one, okay, because we're saying that those in the same family are more closely related than those that are only in the same order. That seems possibly reasonable. And then we have family genus, we have kingdom phylum, and we have class order. Notice all of these, they're going top down, top down, top down, except for the one, it goes down, up, right? The one that goes up is going to be our answer here, because if they are in the same category lower down, more specific, that means that they're more closely related than the more general categories, right? You guys get that? All right. The second question, and stop me at any point if what I'm saying is not jiving with you, okay? Our second question is about chemistry. Yay. What is the maximum number of covalent bonds that one oxygen atom can make with one hydrogen atom? If you were with Professor Thomas, what was something that she taught you guys to remember this? Honk. So it's just a fun little mnemonic here. Honk. 1, 2, 3, 4. Okay? What this means is that that's how many bonds each of them are capable of making with another atom. Okay? So carbon can make up to 4 bonds. Nitrogen can make up to 3 bonds. Oxygen can make up to 2 bonds. And hydrogen can make up to 1. Right? Does this make sense? So, what we're looking at is we have an oxygen and a hydrogen. We want to know how many bonds can be made between them, okay? And this is tricky because oxygen can make 2, hydrogen can make 1. So what do you think our answer will be? It's going to be 1. Because we're limited by what the hydrogen can do. We can't make the hydrogen do more than it's capable of just to meet oxygen's demands. Okay? However, if I had like another hydrogen, I could slap it on over here on the other side. You know what I'm saying? So, but in this particular question, we're saying 1 oxygen, 1 hydrogen. That's the idea. Okay? Again, stop me if you want to. Okay. Nitrogen is much more electronegative than hydrogen. Which of the following statements is correct about the atoms in ammonia? Okay? So, we have ammonia, nitrogen, and then some hydrogens. And you don't have to be able to draw this for your exam, but this is just kind of to visually see what's happening. We're saying that nitrogen is more electronegative, okay? So that means that there's going to be like a pull this way. And the nitrogen is going to take on a partial negative charge. What molecules does this seem very similar to? It seems really similar to water, right? But we just have a different one that we're working with. But the principle stays the same. That's the idea, okay? So if the nitrogen is pulling all the negative, then what happens to the hydrogens? Yeah, so they're going to get a partial positive, right? Plus, plus, plus. So you see how there's a big pull on one side And then, so it's negative on one side, positive on the other side. What do we call this type of molecule? Polar. Excellent. That's a polar molecule, okay? Now let's analyze our answer choices. So, nitrogen atom has a partial positive charge. Is that true? Nitrogen, partial positive? No. Does the nitrogen have a strong positive charge? No, that's not, I mean, it's going to be negative. So whether it's strong or weak, it's not going to be positive, okay? There are covalent bonds between the hydrogen atoms. So that's a tricky one because you might be thinking, well, maybe. But if you're going to have the hydrogens connected to each other, think about this. So you're going to have hydrogen, hydrogen, hydrogen. We can't have two bonds on a hydrogen, right? So this is not correct. Okay? And then the last one, or not the last one, the next one, each hydrogen atom has a partial positive charge. Yeah, you guys are thinking that? All right, we'll flag that for evaluation. And then each hydrogen atom has a slight negative charge. No, so we're going to go with D. But yeah, this is really getting down to the concept of like polar, nonpolar. Yeah. That's a good question. The question is, why is it a partial positive charge? The answer is because it's only leaning that way. In the sense that they're still sharing the same electrons. It would be a strong positive charge if it was like an ionic compound and they had reacted in that sort of way, where one had given electrons to the other, but because they're still sharing them, it's only a partial. Yeah, thank you for that. Okay. Also, if any of you guys over here are trying to ask me stuff, do something to get my attention, because when I'm looking at the paper, this computer is obstructing my vision, and I can't really see you. So make sure it's very clear if you got questions for me. All right. Yeah. Well, the hydrogens wouldn't have a partial negative. The hydrogens would not have a partial negative. You know what I'm saying? Yeah. No, so I guess I'll answer something not quite exactly what you'd asked, but something related. I can't imagine that you would get answer choices that were like partial and slight and having to compare between those, because that's the same thing. So you would get one or the other or something different. Yeah. But, I mean, you can ask me, maybe come talk to me. Oh, instead of slight being partial? Yeah, they're synonymous, so you wouldn't have two answer choices that were the same like that. Yeah, okay, we're good. All right, any other questions on this one? Okay, let's keep going. Okay, now we're talking about covalent versus ionic bonds. This is kind of important. Okay, what do we know about covalent bonds? Huh? Yes, thank you. They involve the sharing of electrons, okay? Covalent bonds, we're sharing, right? It's that friendship. We can either share equally or we can share unequally, but we're sharing. What about ionic? Do we know? Huh? Yeah, they're like transferring electrons to each other. And then they're attracted after that because they transfer those electrons. So one becomes like permanently positive, one becomes permanently negative. Okay? All right, so let's take a look at our choices here. Covalent bonds involve the sharing of neutrons between atoms. What do we think of that? No. Since when do we share neutrons? I don't think so. Covalent bonds involve the sharing of protons. Nah. Covalent bonds involve the transfer of electrons. Yeah, so we do finally we do finally have electrons here, but covalent bonds don't involve the transfer, right? So that's not going to be okay. Covalent bonds involve the sharing of protons. Again, we got a hit. Covalent bonds involve the sharing of electrons? Okay, that seems reasonable. So finally we have like part of a choice that looks good. Now let's follow up with the second part. And ionic bonds involve the electrical attraction between atoms. Does that seem okay? Because after one gives its electrons to the other, one of them becomes positive, one of them becomes negative, and they're electrically attracted to each other. So that's the idea here. Okay? E is our answer. Okay. So, this next one, we are talking about some other stuff. We're kind of moving through here. Which of the following molecules orient themselves into sandwich-like membranes because of hydrophobic components within the membrane? You guys know? D? Yeah, you guys remember? We're talking about membranes, right? When we talk about membranes, what makes up membranes? Phospholipids. And we can take a look at them, if you recall. Remember, they have this kind of thing. Right, so they kind of look like that, right? It's this double layer. Remember we called it the phospholipid bilayer? No? I think I heard someone say no. So we have, these are the polar heads, and then these are the, that's the same thing, and then these are the non-polar tails. You guys remember that? So because we have this polar and non-polar situation going on, stuff is able to pass through in a controlled way. You guys remember that? Hopefully? Well, that's the stuff you want to remember, okay? Let's check out this next one. Hydrophilic properties are characteristic in all of the following except what? Okay, so what does it mean to be hydrophilic? Hydrophilic means that we like water. What kind of things like water? Huh? Things that are soluble in water. So are we thinking polar or nonpolar? Polar, nonpolar. What kind of things like to interact with water? Polar. I wanted a more resounding answer than that. All right. So this just kind of like, you know, we get this question and then we start brainstorming a little bit and then we kind of work towards an answer, okay? So we know that polar molecules are probably going to be hydrophilic, right? Molecules soluble in water. Does that seem reasonable? Hydrophilic as well. That's when we say it likes water, what would be something that does not like water? Oil, right? You ever go to make a salad dressing? You can't just like pour the oil in there and stir it and hope for the best. You have to like very carefully emulsify it, and then you walk away for a couple minutes and you come back and it's all separated. So, anyway. Molecules that readily ionize in water. What do you think of that? Yes, maybe? No? We're not sure? Well, I'm gonna put a little thing here because I didn't get a very strong response. And then we have the long hydrocarbon chain components of some molecules. Now, when we talk about hydrocarbons, what are we normally talking about? When we talk about long stretches of carbon and hydrogen over and over. Huh? You guys remember? Yeah, the tails of phospholipids, or even, like, that's just fats in general, right? So fats, oils, nonpolar, right? You gotta get this thing. But also talking about, like, the tails of the phospholipids. It's the same kind of deal. So that one is looking a little suspicious for being hydrophilic. But let's check this one again. So if we're saying molecules that readily ionize in water, if they form ions, like if I do this, does that seem like it has some degree of like a polar type nature, right? Even though it's ionic, normally when we say polar, we're talking about covalent compounds, right? But ionic compounds are still going to be hydrophilic because they have that charge difference, and that charge difference is going to make them attracted to the water, the different sides of the water, right? So we're going to go with this one, right? Because this is fats, that kind of thing. Yeah. Oh, so there is. Allow us to evaluate and make sure that doesn't change our answer. Man, I hate when that happens. Okay. So our last answer choice was a hydroxyl group. What is hydroxyl? You sure it's COOH? No? So that was carboxyl, right? COOH, like it was like this. That's carboxyl. But if we have like COH, that's this guy. So hydroxyl is actually also going to be hydrophilic because it's going to have the same properties that water is going to, where the oxygen is going to pull on the electrons. So the oxygen becomes negative, the hydrogen becomes positive. Good stuff. So, yeah, we're actually going to eliminate this choice as well, but I appreciate you guys bringing that to my attention. I didn't even realize I did that. All right. Any concerns with this one? Okay. All right. If the H plus of a solution is 1 times 10 to the negative fifth, what is the POH? I know you guys are so excited to do these again. So how do we do this? You guys remember? So we have the H plus concentration, okay? And it's 1 times 10 to the negative 5. So can we find the pH? What is the pH? 5. Right? It's negative log of the concentration, but for all of these problems for Bio 1, you just take the number from the exponent and you're done. So how do we get to POH? Yeah, so pH plus POH equals what? 14. So POH will be 9? Something to think about for these, because this is the only pH problem I put on here. Don't forget that pH and POH, you're looking for a number between 0 and 14. But if they ask you for the H plus or the OH minus concentration, then you're going to be dealing with a number with an exponent. Okay? So just make sure that you are aware of those distinctions. Questions here? All right. The next one, how can peptide bonds within a protein be broken? So this is a tricky thing, right? We're talking about peptide bonds. What are peptide bonds between? What are the pieces that make up proteins? Amino acids, right? So remember, let's do a little review real quick. So for an amino acid, we have carbon, and then it's attached to an amino group. And then we have a carboxyl group. Then we have a hydrogen. What's the other group? R group. What does that mean? What do we mean by R? It's the variable group. right? It means that it can be anything, or rather, any of the amino acids. That's what changes between them, is whatever the R group is. So remember, you guys were responsible for learning that part? Okay, so when we talk about peptide bonds, we're talking about the bonds between two amino acids, right? And if we're making proteins, what kind of molecules are proteins? You guys remember? Well, this is a chapter 5 question, so that's a hint for you. So what kind of molecules are proteins in general? Macromolecules, yeah, chapter 5, right? How do we put together and take apart macromolecules? Yeah, so so these answer choices are meant to sort of throw you off a little bit. The denaturation, remember we talked about you can denature a protein, but really that's going to just take it back down to the primary structure, right? You have primary through quaternary, potentially. So the primary structure, though, is where we have the peptide bonds. Every other level of structure has the hydrogen bonds. The peptide bonds are made the way any monomers are assembled into a polymer, okay? So when we are assembling a polymer, we do dehydration. Sometimes it's called dehydration synthesis, but I wanted to leave that out to be a little bit more vague. But that's how we put them together, so that's not going to be the answer. How do we take them apart? Hydrolysis. And E is not true. I can't think of anything in biology that is 100% permanent. Everything is able to be undone. So, remember, hydrolysis, you add the water to split them. Hopefully remember. I'm going to take your overall silence as memory. All right. The cellular structure that is involved in producing ATP during aerobic respiration is... You guys got this. Yeah. Don't overthink this now. ATP, cell respiration, mitochondria. Alert. This is a thing. We go with this? You guys remember, we were all there, chapter 9, fun times. Okay. So again, talking about producing ATP during aerobic respiration, that's the key. All right? Let's let's take a look at a couple of these other ones here, okay? What does the nucleus do? Nucleus has DNA, right? What about the nucleolus? You guys remember? This should be very fresh. Yeah, this is where we make RNA. What did chloroplasts do? What's that? They make sugar, right? Is in plants? Okay. They make plants, and then, they make plants. What am I doing, guys? They make sugar, and this is in plants. Something's happening. All right, mitochondria, that's where we make our ATP, the powerhouse of the cell. You guys remember. But I worded it differently, and that's what threw you off. And then we have the endoplasmic reticulum. What kind of stuff do we have? We have rough and smooth, right? What's in the rough one? We got ribosomes. What do ribosomes make? They make proteins. And then we got smooth. What kind of stuff do they do? They make lipids and things. Yeah. All right. I just want to make sure we remember this stuff. Okay. Which of the following cellular structures are common to both prokaryotes and eukaryotes? Ribosomes. I can't even stop you right now. So, what is it that prokaryotes do not have? Well, yeah, they don't have those, but in general, what do they not have? They have no, what kind of organelles? Membrane. Does this look familiar? They don't have any membrane-bound organelles, right? So, bacteria, you got to think, what do they have? They have a cell membrane. They usually have a cell wall. There's like one that I can think of that doesn't. They have DNA, they have RNA, they make protein, so they have ribosomes. But they don't really have any of the other stuff that we talk about, right? Lysosomes, peroxisomes, vacuoles, mitochondria, Golgi, ER. They don't have any of that. So they're just kind of doing their thing, living the simple life. All right? Okay, so this next one, we're talking about the plasma membrane again. What does it primarily consist of? Yeah, so C looks like a pretty good choice here. We have proteins embedded in a phospholipid bilayer. So again, we have, if I bring back my little picture that I drew, I'm going to put it on here real quick. You guys can see, remember this is my membrane, and then you're going to have like some kind of protein, then you'll have more of them, right? What is the purpose of the proteins that are inside the membrane? Yeah, so transport, I mean, there's lots of functions, right? Usually we're talking about transport, so we talk about like carrier proteins, channel proteins, those kind of things. We have like signal transduction that happens. So there's reasons to have these proteins, right? Remember the transmembrane proteins? All that? Okay. Are we good with this one? Okay. All right, now we're talking about movement of water. When we're talking about movement of water, let's see here. When the concentration of solutes differs on the two sides of a membrane, permeable only to water, we want to figure out what is water going to do. Okay? What's going to happen? What do you think? Yeah? E? So let's take a look. Okay, so I'm hearing A and I'm hearing E. Okay? I'm hearing A, I'm hearing E. Guys, come back to me. Okay, so we're going to say B, C, and D are probably not the answer. We all good with that? These are gone, and then we're down to A and E. So either water will move across the membrane or solutes will move across the membrane. Which one is it going to be? Well, we say the membrane is permeable only to water. What does it mean to be permeable? It means that it allows things to pass through. So if it's only permeable to water, then are the solutes able to move? No. So even though, so E sounds like a really compelling answer choice, and you're probably like, yes, I'm going to put this, it's really long, it has all these words, it sounds really compelling. And then also it says solutes will move across the membrane from the region of higher concentration to lower concentration. That would all be true if the solute could move. So in these questions, you have to think, what is allowed to move? And then that will move from its higher concentration to its lower concentration. Bless you. But because we said solutes cannot move, it has to be A. Yes? So we could do something like this. Let's do Um, if, so we'll say, if, to, glucose and water. So then we'll just say, we'll make this kind of simple. We'll just say, what is able to move and why. Okay? So then what we'll do is we'll create our membrane, and then we'll say we have like lots of solute. Okay, so the dots are glucose, okay? This is all my glucose. All right, and so you see that there's more glucose on one side than the other, right? So the first thing that's going to move would be your glucose. So it's going to move across to the other side. But it's only going to be until it's all even, right? So we're going to end up with equilibrium on both sides, okay? So the glucose is going to move until it reaches equilibrium, which means until we have the same amount on both sides. And then the water is going to move to wherever there's more solute. You guys catch what I'm saying? So if, based on this move, so we would need more information for like a real question, but if this move means that there's now more solute on this side, then the water will follow it. If this move means that there's more water on the other side, the water will go that, or there's more solute on the other side, the water will go that way. But the idea is you can only move solutes if the question says that you're allowed to. I don't know if you guys recall, I have posted, I think it was this was from the second exam review, right? Where we did chapter 7. Remember I made that like YouTube thing? Like not like Y-O-U, but literally U. And we moved glucose, and then we didn't move sucrose or something like that, and then the water changed. You guys should take a look at that one again, because that's a good example of this happening. Okay? Let's go back. Can I define osmosis? Yes. Osmosis is the diffusion of water. I'll write this down here. So it's either the diffusion of water, or you can just simply say the movement of water from a high concentration to a low concentration. So it's just like anything else, but specifically the water moving. The reason that this is a big deal is usually, you'll have membranes that are only permeable to water. The reason that you should care is because if you end up in the hospital, which I'm hoping that you don't, but if you end up in the hospital and you need fluids in your body, right? They find out that you're dehydrated. If they give you a solution that is different from the solution that's already in your body, something's going to happen. They're either going to pull all the water out of your cells and kill you, or they're going to add a bunch of water to your cells and probably kill you. So, or cause you severe harm until it's corrected. So that can be a real concern. Yes? Yeah, that still applies. Sorry, the microphone's over there. So we had spoken in a previous review about how while on the one hand, you can say water is going from its higher concentration to its lower concentration, the water is also moving towards wherever there's more salt to like dilute it. So it's kind of like trying to like water it down in a way. So bless you. We can actually take a look real quick because we have plenty of time here. So we can do something where we say that like if a cell has 20% salt in a solution with, with, I don't know, 30% water, and the cell is only permeable to water. I don't know what you want to say to the cell. We can say what will happen to the cell. There's like a million different ways you can make these questions, but we'll do it like this, okay? So in this particular situation, like it's usually a good idea to draw this out. So we'll make our cell here. The cell has 20% salt. So how much water does the cell have to have? 80%, right? We're assuming it's got these two things. Okay, and then the solution has 30% water. So then how much salt does it have? 70%. Okay, so we're going to analyze this based on two perspectives. First perspective being movement of water, right? So if we say that the water wants to go from its higher concentration to its lower concentration, where is the water going to move, in or out? So is it going to move in the cell or out of the cell? Out of the cell, right? Because there's more water in the cell. The other way to think about it, the one you were just asking me about over there, is the idea of like trying to dilute the stronger areas. So if there's 70% salt on the outside, 20% salt on the inside, that means that the outside is like really, really salty. So you want to think about it like it's trying to make everything equal, so it's going to like go try and water it down. That's the idea. But again, the water's going to leave. So whatever works for you. So the cell, well, there's a couple things we can say. The cell will shrink. Okay, so that's one way of saying it without getting into the terms, but now we can talk about the terms, okay? So what kind of solution is this cell in? So we can say the cell is in a hypertonic solution. Or, if you want to say it another way, you can say the cell is what to the solution? Hypotonic to the solution. All of these statements are true. Okay, so whichever way you want to look at it. But one good way to think about this, hyper in like medical terminology means more of something, right? So if it's hypertonic, that means that it has more salt. So the solution is definitely going to be hypertonic here, okay? So this would be the equivalent of, again, you end up in the ER and they go to give you saline, but for some reason it has too much solutes in it. It's got too much sugar or whatever. And so then all of your cells start dehydrating. This is what happens if you ever see, like, you know, they say you shouldn't drink ocean water. There's a reason why you shouldn't drink salt water, because this will happen to you. So in a cruel twist of irony, if you're stranded on a deserted island and you're so thirsty, and you drink the salt water, then it's going to make you thirstier or kill you, depending on how much you've had. So it's not a good way to go. I don't recommend it. Okay? All right, let's move on. Okay. This one, I know we have more choices. Okay, so this one, this particular question, we're talking about the smooth ER, okay? So the smooth ER exhibits all of the following activities except what? What do you think? You think A? Why not A? Yeah, because the rough ER makes the proteins, right? So even if you don't remember everything it does, let's say I'm gonna be in my exam for a moment. Okay, I know it makes lipids. I know it breaks down toxins and toxic byproducts, right? I know it detoxes stuff. But let's say I forget that it makes hormones. That's okay, because if I know that the rough ER is going to make proteins, that's all I need to answer this. Okay? Okay, so ATP is required for all of the following processes except, okay? So we're trying to figure out what's going to need an input of energy. If we're doing active transport by transport proteins, does that need ATP? Yeah. If we're doing facilitated diffusion, yes, no, I've heard a mixed bag. I'm going to flag it. What about microtubule movement? You think so? We're moving stuff? What about sodium-potassium pumps? So even if you don't know what a sodium-potassium pump is, it's a pump, right? So what do you think pumps do? They're going to push things, right? We got to force it somewhere, okay? And then what about protein synthesis? You guys remember translation, right? I know it's been like a couple days. So translation, all those things we did, the small ribosomal subunit, and then the mRNA comes, and then we have the tRNAs and the large subunit, and everything moves. You think that all just happens, like, passively? No, we have to put so much energy into it, but it's okay because we like protein, so we're going to make the effort, okay? But that is gone here. We're going to go with facilitated diffusion because facilitated diffusion is going to be passive, but with the aid of proteins. Okay? So it's a type of passive transport. That's the one that has the carrier and the channel proteins as options for it. Do you guys remember that? We feel good about this? I'm hoping this is just helping like bring back these memories and kind of rekindle these thoughts for you. Okay. The next one, which one of the following is an anabolic reaction? So now we're going to take a time out for a second. Before we analyze our answer choices, I need to know what is an anabolic reaction. So it's going to need energy. And is it going to build things or break things down? Yeah, so it's going to build things. Okay, so we're kind of armed with a little bit of a strategy here, so we know where we're going with these choices. I really recommend that when you get to a lot of these questions, don't go fishing for answers in the answer choices. Your exam questions are designed to have compelling wrong answers to make you pick them, right? The wrong answer choices aren't just going to be like, grape or something. Like, it's going to be things that are reasonable for you to pick. So you want to actually have an idea of what you're doing before you look at your choices, okay? So Let's take a look. So we have ATP plus H2O makes ADP plus phosphate. Is that anabolic? What is happening here in that reaction? Do you guys know? So this is going to be catabolic. We're breaking this ATP down into ADP and phosphate. That's us using the ATP, right? We've just used this energy. So that's the actual use of it. So that's not going to be anabolic. Okay, so then we have this thing. B, what is choice B? Does this look like something you've ever seen? Yeah, so choice B is cellular respiration, and choice C is photosynthesis. True story. Okay, so what is the purpose of cell respiration? What do we start with? We start with glucose. We start with a sugar. What is the desired output? ATP, right? So overall, are we having an input of energy to build something in this process? No, we're breaking something down to get energy. We're trying to get that ATP out of it. So cell respiration is not an anabolic process. All right, what about photosynthesis? What do you start with in photosynthesis? If you have a plant at home, how do you take care of it? You give it water and sunlight, right? So it starts with energy, right? And then we are building a sugar. That's how this happens. So that is going to definitely be an anabolic process. Glycolysis is part of cell respiration. Everyone good with this one? Okay. Okay, now we're going to talk about enzymes for a moment. So, enzymes have all of the following properties except what? So you got to think, what is an enzyme? What's the purpose? Do enzymes increase the rate of a reaction? Yeah? Right? Enzymes make reactions happen faster? Yeah. Do enzymes lower the activation energy? Yes, we remember that. And that's how it makes them go faster, right? Like, let's say, all right, guys, I have a perfect example for you. Anyone have to, write any papers or anything for the end of the semester? Yeah, a couple of you guys? Okay, so let's say that your paper is supposed to be 10 pages long. I've done that a couple times. It's really a bummer. So you have a 10-page paper. That's going to take you a while. If I was your professor and then I sent you an email and I was like, hey, just in case you haven't finished your paper yet, it only has to be 3 pages. Would that increase the rate of your paper production, right? If I make it easier for you to do it, you can do it faster. That's the idea. If I give you all the answers to your exam, you're going to probably answer them faster. That's the idea. So it lowers the energy needed to overcome to do this reaction, so it goes fast. Okay? Do enzymes have specificity for certain molecules? So what this means is, are enzymes specific to certain things, or does any enzyme go with any reaction? They're specific. Yeah, otherwise we wouldn't learn all those DNA enzymes, right? If any enzyme could do anything, then we could just have DNA polymerase do the whole show. But we can't. So that's gone. Are enzymes going to increase the amount of products made? What did we say enzymes cannot alter? So enzymes cannot change what? You guys remember? I heard it. Delta G. Delta G is the overall energy of the reaction. So enzymes cannot change what will happen. They just change how fast it's going to happen. You know what I'm saying? So that one is not going to be valid. That's going to be our answer. We were almost there. We almost had an all of the above. Okay. Questions here? We still good? All right, we are still good. What is the purpose of oxygen in aerobic respiration? Okay, you guys remember this, right? We talked about it many, many times. So, oxygen accepts electrons at the end of an electron transport chain. Does that seem important? Yeah, maybe? Put a little dash here. Is oxygen necessary to carry away CO2? Is that what we form with oxygen? At the end? What do we form with oxygen at the end? Water, right? So that's not it. Oxygen is used in the formation of sugar molecules. Are we making sugar molecules? No. The oxygen molecule becomes part of the ATP molecule. What do you think? Not so much. Does oxygen donate H+ used in the formation of NADH? It always comes down to this. Oxygen is your final electron acceptor. That is the most important task for oxygen. Technically, B is true to some degree, but it's not the primary reason that we need oxygen. The most important reason is because we need to get those electrons out of there at the electron transport chain, right? This all kind of connects. Okay. Questions here? All right. All the following processes produce ATP, except for what? Does glycolysis produce ATP? Yeah, we make 2 net. How many total? Yeah, you guys remember this. 4 total, 2 net. 4 total, 2 net, right? Does the Krebs cycle produce ATP? Yeah. Does lactic acid fermentation produce ATP? Let's say you have a giant question mark in your brain, so we're going to just put a line next to that. What about oxidative phosphorylation of NADH? Now, what do I mean by that? When do we have oxidative phosphorylation occurring? In the electron transport chain. Do we get lots of ATP out of the electron transport chain? Yes. So we have NADH and FADH both going to the electron transport chain. Our only choice left is C, which is lactic acid fermentation. By the way, if you're at all curious, the purpose of lactic acid fermentation, you ever decided one day, I'm going to be a runner today. I've done that before. And so you go out there and you're like, this is going to be great. And you're running and you're running and you're running. And eventually you start to feel the pain. Assuming you're still breathing, your legs probably start to hurt, right? You feel that pain after you're running? So you're actually producing an acid in your muscle cells because you're not able to do cell respiration fast enough because you're like, whoa, I need way more energy than I was ready for today. I didn't think we were doing this, body. So what happens is, you go through glycolysis, but you're not getting enough oxygen to your cells. If you're not getting enough oxygen to your cells, you can't do all of cell respiration. So we do this fermentation process to be able to keep doing glycolysis over and over without oxygen. Because remember, glycolysis is anaerobic, right? Remember that? I'm going to assume you do. Okay. Chemiosmosis describes how ATP is generated from ADP. What is chemiosmosis, other than a way that we generate ATP from ADP? When do we do chemiosmosis? Electron electron transport chain, right? Yeah, so this is another oxidative phosphorylation and chemiosmosis are, for all intensive purposes, two ways of describing very similar processes. Okay, and they're both occurring at the electron transport chain. All right? So we're trying to say all of the following statements conform to the process as it occurs in mitochondria except for what? Okay? So we're just basically trying to figure out which one doesn't seem right. Does H plus accumulate in the area between the membrane of the cristae and the outer membrane of the mitochondrion? Yeah? Maybe? Yeah? Okay. So we're going to say, we're going to get rid of that one then. Do electrons travel along an electron transport chain? Possibly. Do NADH and FADH become oxidized? So that's the thing, right? We got to try and remember what oxidation and reduction are. Let's take a knee for a second. Let's figure this out. Okay, so when we have NADH, okay? When NADH shows up to the electron transport chain, what does it do? It drops off those electrons, right? So we end up with some stray electrons, and then we have the rest of it, the NAD+. Okay? So what we need to do is we need to analyze, is this an oxidation process or is this a reduction process? Okay? So I'm going to give you my favorite mnemonic, which is Leo, the lion, says grr with an E. All right, Leo says grr. So when you lose electrons, you are oxidized. You gain electrons, you are reduced. Okay? The question now for us is, did NADH gain electrons or lose electrons? It lost electrons, right? Going from here to here, it lost an electron, okay? So was NADH oxidized or reduced? If it lost electrons, it was oxidized. Which is why, by the way, if you're at all curious, we call it oxidative phosphorylation, because we start with an oxidation event and we end up phosphorylating ADP. So, there you go. There's the big picture. So, let's come back to our question. Do NADH and FADH become oxidized if they're dropping off electrons? Yes. So, this last one, we're going to take a look. Electrons flowing through the ATP synthase channel protein provide the energy to phosphorylate ADP to ATP. So that sounds pretty compelling, right? But obviously there has to be an issue if we eliminated all our other choices. So what is it? Are electrons going through ATP synthase? It's the H plus ions, the protons. If that word was changed to protons, or if it was hydrogen ions, that would be correct. But it's not. So it's our answer, which is how that works. You always got to watch out for those answer choices that are like really good sounding, and then there's the catch. You got to look for the catch. That's how they catch you. All right? No? Yeah? Okay, and now we're looking at cell division. Hooray. If a cell, whew, guys, I don't know if I'm going to be able to talk at the end of this. If a cell has 46 chromosomes at the beginning of meiosis, and you're like, oh no, not this again. Okay, so we have meiosis. We want to know what happens at metaphase 2. How many of what would we have? Okay? I'm going to redraw one of our little cheat sheet things. Okay? So we'll do this. You guys remember doing this? Okay, so this is meiosis 1 at work, and then this is meiosis 2. Okay? So the purpose of doing this is to look at what happens, right? Once we get past meiosis 1, oh no, I turned on this light. I don't think I like this light. Get out of here. Breaking the thing. Okay, so between meiosis, or between before and after meiosis 1, what change do we have? Okay, are they diploid or are they haploid now afterwards? They are haploid now, as opposed to being diploid here. All right, we always start diploid. But now we have half the number of chromosomes, all right? So that's the difference going from the start of meiosis to the end of meiosis 1, okay? And then are they replicated here or not in the first one? Replicated, yeah? Remember, replicated means we have 2 sticks. They're in pairs, okay? So are we replicated here in one of these? Yeah, they are replicated. They're haploid but replicated, okay? That's a very important distinction. So, again, when we look at, like, that's a chromosome, and that's a chromosome, but that one's replicated and that one's not, and that one has 2 chromatids. You guys remember all this? All right, so then after meiosis 2, are they diploid or haploid? Haploid, and then replicated or unreplicated? Unreplicated. Awesome. All right, so now we need to look at our answer choices and see what happens. So the cell has 46 chromosomes to start with, okay? That means that after, so we're talking about metaphase 2, so we're done with meiosis 1, okay? Do we have the same number of chromosomes that we started with? No, so I'm going to write what we're looking for. We are looking for 23 replicated chromosomes, right? Because we're like right here. So we've cut the number in half, but we're still replicated, right? So that's what's happening. Okay, so now we need to find an answer choice that is consistent with this. Do we have 23 chromatids? What do chromatids mean? They're the sticks, right? We should have how many chromatids? 46. Okay, so that choice is no good. So I'm going to just get rid of our top sheet here. All right, do we have 46 chromosomes? No, again, we're looking for 23 replicated in some way, shape, or form. Do we have 46 chromatids? Yeah, that sounds pretty good. And then 92 chromosomes, 92 chromatids. So 92 chromatids we had at the start of meiosis. 92 chromosomes we will never have. So that's not happening, that's not happening. This is our answer. Huh? Between replicated and unreplicated? Oh, chromosome and chromatid? Well, so this is a chromosome, and this is a chromosome. But this chromosome has 2 chromatids. A replicated chromosome like this one has 2 chromatids per chromosome. That's the difference, okay? All right, excellent. Let's carry on. All right, the ability to taste a chemical called PTC. Did you guys ever do this like in high school? They give you a little piece of paper. It's like kind of bitter. The ability to taste a chemical called PTC is inherited as an autosomal dominant allele. What is the probability that children descendant from parents both heterozygous for this trait can taste PTC? All right, so we're going to try and solve this. So we're saying it's an autosomal dominant allele, okay? And if parents are both heterozygous, I'm going to just do A's here. So our cross is big A little a times big A little a, right? Let's do a box. Okay, so now that we have done that, what we need to figure out is what is the probability of these children being able to taste this, having this trait. You're going 1/4? No? 3/4? How do we distinguish this? So this is an autosomal dominant trait. Normally, when we're talking about traits, we tend to focus on like recessive things because a lot of diseases are recessive. But we're looking at an autosomal dominant allele. So any of these three with a capital A will have it. So it's going to be 3/4 because it's a dominant trait that we're following. Okay, this next one is talking about fruit flies. Okay, so we have dumpy wings, and then we have normal wings. The allele for normal wings, capital D, is dominant to the allele for dumpy wings. 2 normal winged flies were mated and produced 300 normal winged and 100 dumpy winged flies. So we want to know what were the parents. So we have to think about that. No, I said we have to think about that. So let's evaluate our circumstances here, okay? So both of them have normal wings. So each parent, what choices do they have for genotype? So they have big D, or big D, little d, right? Those are their only choices. But both of them have to be one of these, okay? So we can get rid of choice E. We can get rid of choice D. Okay? You guys see how we do this? And then they produce some recessive ones. So could it be A? It can't be A. They have to have access to recessive. So the question is it this one or this one? So let's do it. Let's take a look real quick. Okay, so in this case, we're going to end up with half of them. So half of them are going to be homozygous dominant, and then half of them are going to be heterozygous. What will the output be? So they're all going to be normal winged. You see that? If this is what we have, they're all going to be normal winged. The only way to get 300 normal and 100 with the dumpy wings is going to be if we have access to make it. So it's going to have to be C. Yeah, see how we do this? Okay. We want to know for this next one, which of the following is true of the gametes produced by an individual with genotype big D, little d? What do you think? So What does this mean if that's your genotype? How does it work in meiosis? Are you going to have half of your gametes be big D, half be big D? No, so A doesn't look too good. You want half big D, half little d? Possibly. We'll go with an ambiguous answer here. Do we want half big D, little d, half big D, little d? When we do these Punnett squares, we have a reason for splitting them up. What you're doing is you're saying that half of my children, I'm giving this letter. Half of my children, I'm giving this letter. You're splitting them up. That's why you're doing this. Otherwise, you would have to have a box, and you'd have to be like this. You see? And we don't do that. That's not a thing that we do. Okay? So, that's not an option. That's not an option. That's ridiculous. It's B. Yeah, see? All right. You ready to do a Punnett square with like 500,000 boxes? No? Okay, fine. Then we won't. Okay, so we are going to do a probability here. We have 4 traits, which can be a little upsetting, but this is okay because we're going to, instead of drawing this whole thing out, we're going to use probabilities in order to get us there. So the key is that you want to find the probability of each letter happening on its own, and then we're going to multiply all our answers together, okay? So I'm going to rewrite the one that we're trying to make here. Okay, so if we're crossing big A with big A, what's the probability that we get big A? 100%. So I'm going to put a 1 because that's, with fractions, that'll be how that works. And then big B, little b. What's the probability that we get big B, little b? It's 100%. Because if you think about it, it's going to be big B, little b, right? So it'll be 100% again. The next one, we're going to do big C, little c. This one we're going to get half and half. You see that? Because that's the same thing as this one. So this will be 1/2. And then this last one, big D, little d, that one is going to look like this one. So what's the probability of getting heterozygous? 1/2. So what's our answer? That's it. Yes. The whole thing? So I'm trying to take shortcuts here. But to get the 1/2, we're taking the probability of getting big C, little c based on the parents. So we're doing the cross of big C with big C, little c. Probability of getting this, because that's the goal. Yeah. Everyone good on this? We're trying to find this based on them. Okay. All right, this question. We have, in domestic cats, 2 alleles of a sex-linked gene code for hair color. One allele codes for orange hair, the other codes for black hair. Cats can be all orange or all black, or they can be calico. Hey, look at that. A coat characterized by randomly arranged patches of orange and black hair. I will tell you, if you're in Professor Thomas's class, you will not get that fun little intro. You are expected to know calicos because she is quite fond of them. If you're in Dirkson's class, then, at least to my knowledge, you're not expected to just know the inheritance of calico cats. So you would be given something to this effect, okay? All right, so what are we trying to figure out? which of the following are true? Or all of the following are true except, okay? So, we're going to look at these options, okay? If we have a black female cat, okay, so it's going to be XB, right? Black female times an orange male, okay? So the orange male, he only gets one X, right? So can he be XO? No, it's just going to be XO and then a Y. So, do they have the ability to produce a black male cat? You can actually figure this out without doing the cross. Okay? Let's take a look at this. So, if they're producing a male cat, which one is going to make them male? Yeah, the father is going to give the Y chromosome. Agreed? So we're not asking what's the probability. We're saying, is this possible? Okay? Which is why we don't need to do a box. We're just looking at, can we do this? Okay? So we're going to pull the Y from Dad, and then we're going to pull one of the B's from Mom. So is this possible? Yes. So we get rid of that choice. You see how we're doing this? All right, next one. We have another black female crossed with an orange male. Okay, they want to know, can we make a female calico? Okay, so if it's female, Dad's giving Y, and then Mom's going to give one of these. Calico? Yes. All right, let's see a calico female. XBXO crossed with a black male, XBY. Okay. So we want to make a female calico again. So we're going to pull this X because it's going to be female. All right. And then do we have the ability to make a calico? Yeah, because we can pull this O. Again, I know you're probably thinking, we could pull the XB from the mother, but we're not asking what's the likelihood, we're asking can we do it? Yes, we can. So that one goes. All right, this one, I'm going to start working down here because I'm running out of room. So we're going to have XBXO crossed with XOY. Can they produce a female calico? We pull that from Dad, pull that from Mom. Yes, we can. You see that? So, then this last one, we're taking a calico female and then an orange male. Can they produce a male calico? No. Do you see? I hope you're not upset with me for taking you down this path. Do you see what I've done? So what's the issue? You cannot have a male calico. There's an exception. I'm going to mention that in a second, so just hold on if you're there with me. But a male has to have a Y chromosome, right? And in order to be calico, you have to have a black X chromosome and an orange X chromosome. It's just like saying males can't be carriers for sex-linked recessive disorders. You understand that? So what's the only exception? So, if it is a Klinefelter male, that means that he has two X chromosomes. You have the ability to have XB, XOY. So, if you've ever seen a male calico cat, then you know that that cat is a Klinefelter. Which is kind of interesting. But it's very rare. How do we end up with this situation? What do we call this, this type of thing where we end up with more than one chromosome or one less or something? Something? Yeah? Well, it results from non-disjunction, right? Remember that? All right. Okay. So now we want to know, from which parent or parents did a male with red-green color blindness inherit the defective allele? Okay, let's think about this. So red-green color blindness, what kind of trait is this? Yeah, sex-linked recessive. Okay, sex-linked recessive. So let's think about this. If we have a male who is X little a Y, I'm saying that the little a is our, like, he is colorblind, right? Because he can't have little a, right? If it was a female, we'd have to have this, right, to be colorblind? But this is not happening. It's a male, and we're assuming that he's a normal male, okay? So if he is colorblind, where did he get that defective gene from? Mom. Mom's always. So here's an interesting thing to think about is if you are a male and you have any form of sex-linked issues, it's your mother's fault. So you can let her know that you're very disappointed in her. I'm just kidding. Don't do that. She'll be upset, I'm sure. But yeah, you see how we do that, though? Okay. Alrighty. So this next one, we're looking at a human genetic defect that is caused by non-disjunction of the sex chromosomes. Okay, we just mentioned one, right? Klinefelter syndrome. Okay. Sickle cell, is that caused by non-disjunction? What is sickle cell caused from? Because we learned about this. Yeah, so this is a missense mutation. Remember? We just did this. What about hemophilia? Do we know about that? So that's sex-linked. What about Down syndrome? So this is non-disjunction, but it's non-disjunction of which chromosome? Of chromosome 21, which is not the sex chromosomes. It's a body chromosome. Okay? So that's the tricky business with this question. It's non-disjunction of the sex chromosome specifically. Okay? Turner syndrome, what is that? Yeah, so this is where a woman ends up being, she only has one X chromosome, so we usually write it XO. Does that apply? Yeah, so this is going to be your answer. This last one is sex-linked. You good? All right. Okay, any questions here? All right. Okay. The two strands of a DNA molecule are connected by what? So we have hydrogen bonds. Oh, I'm sorry, I'm kind of dying. We have hydrogen bonds and we have covalent bonds. Is it hydrogen or covalent? Hydrogen, right? The covalent bonds are in the backbone of it, right? So we get rid of both of these choices. Is it hydrogen bonds between codons and anticodons? No. Where do we have codons and anticodons? Yeah. So hopefully all these things are going to sound pretty familiar now because this is what we just did. What about hydrogen bonds between the bases of one strand and the bases of another strand? That sounds pretty good. Let's check out the other one. Hydrogen bonds between the deoxyribose sugar molecules of one strand and deoxyribose molecules of another strand. Not so much. What is that one? Remember, when we have the DNA and it's connected down the center, these are the weak bonds. All right, the strong ones are on the edges. But remember, we open it up with the helicase and all that. We have to have weak bonds in order to be able to break them. Okay. Any questions on that? All right. This next one, all of the following combinations of nucleotides are examples of normal base pairing except for what? I want you guys to take a look at this one for a second. Really think about it. You're thinking C? Ooh, tricky. All right, let's take a look at this. So, can we have adenine from DNA pairing with thymine from DNA? Yeah. Can we have guanine from DNA pair with cytosine from DNA? Can we have thymine from RNA pair with adenine from DNA? Why not? Yeah, so you cannot have thymine in RNA. So that's going to be a really big issue. Let's look at the other choices just to make sure. Can we have cytosine and RNA pair with guanine and DNA? Yeah, so you might be thinking, why would the RNA pair with the DNA? But you remember, that's how we do transcription, right? We match up the base pairs of the RNA that we're making with the DNA that already exists. So that's fine. We're allowed to do that. Can we have uracil on RNA pairing with adenine on DNA? Yes. So we're allowed to do that as well. The only one that we're not allowed to do is C. Okay? All right. So this one. All of the following enzymes are involved in DNA replication except... Thinking RNA polymerase? Hearing a couple people saying that. Let's take a look at the other ones first. What does helicase do? Yeah, so it opens the DNA. And that's definitely in DNA replication, right? Yeah. What does DNA ligase do? Huh? Yeah, so it attaches it all together, right? It connects Okazaki fragments. But this is also during DNA replication, right? Yeah. What does DNA polymerase 3 do? Yeah, so this is the main one. It's going to synthesize the DNA. What does RNA polymerase do? This is going to synthesize RNA. And when does it do that? Huh? Transcription. I don't know what happened with my letters here. I'm going to just try this one more time. And then what does primase do? Yeah, so it adds an RNA primer to DNA. Make sure that you are able to successfully distinguish between primase and RNA polymerase, right? Because they both do make RNA, but it's for very different reasons in totally different processes. So that one is connected to DNA replication. RNA polymerase is connected to transcription. Okay? What are the end products of translation? So, huh? I have yet to weigh in. I have yet to say anything. I'm just getting a vibe from the room. All right, who's voting A? Who's voting B? Anyone for C? Anyone for D? How about E? All right. So it looks like we don't like C, D, and E. So we're between A and B. Now, a lot more people voted for A. Why would you pick A and not B? Yeah, so the whole purpose of translation, we're not making amino acids. They're already made, but we're assembling them together, right? So it's kind of like if you want to make a necklace, right? So you go to the store and you buy all these different colored beads, and then you making the necklace means you're putting together, you're ordering the beads in the way that you want. You're not actually sitting there melting plastic making beads, right? So that's the difference. So we are going to pick A. Okay? And so this one, believe it or not, guys, this is actually our last question. We finished a lot quicker than I expected. They took off some stuff that used to be on the final, and so it ended up going a little bit shorter. But I will be here for the remainder of the time for any other questions that you have. But hold on, don't get all skittish, because you promised me your time until 5. So give me a couple moments of your time, okay? Listen. All right. An mRNA actively being translated in the cytoplasm would have all of the following except for what? Introns? Why? Because we cut them out, right? So it'll have a poly A tail. It'll have a 5' cap. It'll have RNA nucleotides, we hope, and we're going to keep the exons. We get rid of the introns. Now, give me a moment, please. Okay? I want to just have a second to talk to you guys. All right? So a couple of things. 1, please be sure to do the survey if you're able to on your way out. 2, I'm going to be here for a couple minutes to help you if you have any other questions. And really, thank you so much for everything this semester. It's been really, really awesome. I really appreciate you guys. I won't be doing SI anymore after this, so this is my final hoorah with you. I really, really appreciate it. Good luck on your finals and everything you do. She left.